So, students, welcome everyone. So, I think this is the maximum composition students I think will join us. So, I had some fever in earlier time, but today I took some medicine, so I might not have that good energy today to speak. So, please cooperate with me. I hope my voice is loud enough for all of you. I cannot speak but loud today. Okay, good. Hmm. That is the titles that we did yesterday. Nucleic acid, types of DNA. Types of DNA we have to do still. So let's go one by one. So we did till here yesterday. So let's see what other topics we can manage in this within this topics of today. So I hope my class streaming is good. My voice is loud enough and all in a good go to go today. Good. So what is genome? Actually, every organism, whether it's a mouse, human, ant, Drosophila, Arabidopsis thaliana, all yeast, bacteria, all have our different genome have been identified from the discovery of 2003. Many have been identified till now. So genome is entirety of organisms, hereditary information. This include all the information that is required in order to understand that 1 to 2% of genes that are coding for the proteins in our body, where are they situated, what is their name is. So basically that is the genome is being included. So it is encoded either in DNA or many types of viruses in RNA, and genome includes both the genes and the known coding sequence of the DNA. So if we compare the genome sizes of organisms, as we had discussed, Homo sapiens, that is a human, mouse, fruit fly, plant, roundworm, yeast, bacteria. So for the human, the size is around 3.2 billion and the chromosome numbers are 46. But if we see in the bacteria, the prokaryotes, size is 1.8 billion, but chromosome number is only 1. Also so from the E. coli. So all the bacteria are having 1 1. On the other side, yeast still has 32. Your Drosophila has 8. So that's why Drosophila is a quite good, very nice model if somebody is interested with the genetics. And the mouse and the humans, they are told to be quite similar. As you can see, they have almost similar number of chromosome number. That's why mouse are highly used in the experiments in order to find the disease in human also. So this comparative genome sequence of organisms has highly value, could be used for various things. So why we need genome analysis at the end of the day? So genome analysis is required for the prediction of genes in uncharacterized genomic sequences. In order to obtain the complete sequence of as many as genomes possible, and also for the modifications of a genetic information, so that we can bring out the new varieties like BT cotton, BT brinjal, uh, and many other transgenic crops. This was not in your course at the moment. What are the various tools in the molecular biology? But still, we will discuss few of them so that uh, we are on the good go to go further. So the tools that are used in molecular biology is basically is your PCR, gel electrophoresis, SDS page, Agrose electrophoresis, southern blotting, northern blotting, are the main techniques that are being used. So in the gel electrophoresis, mainly you differentiate between different proteins based on their molecular size with the help of molecular sieve of your polyacrylamide gel, that is PAGE. On the other hand, SDS coagulated and it's get attached towards it, make your protein linear and attached to its whole part and make it negative. So the basic principle, if you want to differentiate between DNA, RNA proteins, they could be separated by the means of electric field. So in Agrose gel electrophoresis, DNA and RNA can be separated on the basis of size by running the DNA through an Agrose gel, and proteins can be separated on the basis of size by using an SDS page gel. So on the basis of size and electric charge, so by using is known as your 2D electrophoresis. So this is your negative electrode, from negative to positive going. So here we can see some negative charge particles. DNA bands are separated by size, and you have DNA bands which are exposed on the films, and then we can see these bands as per the results. Then there is a polymerase chain reaction which is done in order to get the millions copies of same kind of DNA sequence uh, in a infinite way. So PCR has many variations like reverse transcription PCR or amplification of RNA, or real-time PCR, that is qPCR, which allows the quantitative measurement of DNA or RNA molecules. So what happened in PCR? Basically three steps: DAE, denaturation, annealing, elongation. So in the first step, as you can see, you have a template DNA and you want this wanted DNA, wanted gene, this one. So after the first cycle, after denaturation, elongation, annealing and elongation, you will have 4 copies. Then after the second cycle, you will have 8 copies. By the fourth cycle, 32, and by the 35th cycle, it will grow exponentially to make it to 68 billion copies. So basically in the PCR, your DNA replication is used as a principle. Just have a look over this PCR image at how you have many copies of your required DNA leads to the growth of after the 35th cycle 68 billion copies at the end. Now for PCR, we need a primer, which is strand for nucleic acids that serve as a starting point for DNA synthesis, and they are usually short, chemically synthesized oligonucleotides with a length of about 20 bases, and they are hybridized to target DNA, which is then copied by the polymerase. So the minimum primer length that is used in the application is around 18 nucleotides, and the replication starts around 3' end of the primer and copies the opposite strands. So in most cases of natural DNA replication, the primer for DNA synthesis and replication is a short strand of RNA. So some applications of PCR. It is used for the study of patterns of gene expressions. The task of DNA sequence can also be assisted by PCR. Further, for the phylogenetic analysis from the Asian sources, genetic mapping, in order to find the close relatives also. That is parental testing. So now third technique that we can use after PCR and electrophoresis is macroblotting. Macroblotting and probing means mainly your southern blotting that is done for DNA, northern blotting for RNA, and western blotting for your protein. So for the macromolecule blotting, what we do is we having a genomic DNA. We restrict them with the help of restriction endonuclease, digest them with the restriction endonuclease. Then with the help of agarose gel electrophoresis, we separate each particle. And then you have a blotting buffer at the end, and we transfer with the help of onto the nitrocellulose membrane from your agarose gel. And then we probe them with the 32 phosphorus probe, and then we expose to the X-ray film in order to find out the right results. So as I discussed, DNA is basically done for the southern blotting is done for the DNA analysis, northern is done for the RNA analysis, and western is done for the protein analysis. Just a second, students. I just need to go to washroom. I come back. So molecular markers. Okay, sorry, I was out of sight for a second. So molecular markers are basically based on naturally occurring polymorphism that is happening in DNA. So it is done in order to find out the sequence which has been traced to specific locations. So like mini sites, so we have many genetic markers. So like RFLP, AFLP, RAPDs, VNTRs. So restriction fragment length polymorphism, amplified fragment length polymorphism, random amplification polymorphic DNA. Um, so basically then single nucleotide polymorphisms. So these three or four are quite important and we will discuss about them one by one now. So there should be some conditions that have to characterize a good marker, a good suitable marker. That is, they should be polymorphic, codominant inheritance, randomly and frequently distributed throughout the genome, easy and cheap to detect, and reproducible. So molecular markers can be used for several different applications, including germ plasm characterization, genetic diagnostic, characterizations of transforms, study of genome, paternity testing, so like what we have discussed the previous applications. So first is your RFLP, which is your restriction fragment length polymorphism, which is involved in fragmenting a sample of DNA by restriction enzymes, which can recognize and cut DNA wherever a specific short sequence occurs. So as the name suggests, restriction fragment length polymorphism, that is they're cutting from specific point, and then the polymorphism leads to find out the specific things that we are looking for with the help of restriction enzymes for the further genetic analysis. Then AFLP, this is as name suggests, we use amplified fragment length polymorphism. So in this we use PCR in order to find out the results, so that we have amplified versions of our segment. And the third one is your random amplification of polymorphic DNA, which is a type of PCR, but the segments of DNA that are amplified and random. Then this is SSRs, microsatellites, simple sequence repeats, SSRs, STRs. So they're basically the repeating sequence units having 1 to 6 base pairs of your DNA. And this is quite important, SNPs. So they are single nucleotide polymorphism. So if it is GCG, it will change to TA. So your pairing of your base pairs will be changed and leads to the very important part for the transgenic crops, livestock research, or any further breeding programs. And STRs are short random repeats, so some 2 to 6 base pairs for like CAT, GC, they will keep repeating, and they are mainly the non-coding intron regions presence, and they are quite useful in the forensic cases and used for genetic fingerprinting of individuals. So how can you isolate DNA and purify it further? So this could be done. DNA can be isolated from any nucleated cells who has a nucleus, will have chromosomes. And inside these chromosomes, as we had discussed yesterday, will have a coiled beads on a string DNA. And actually DNA is a giant anion in a solution. So what are the various sources of DNA includes? Basically it includes your blood, buccal cells, cultured cells, bacteria, biopsies, forensic samples, that is your body fluids, hair follicles, bones, and teeth roots. So first of all, there are three steps in the DNA isolations. That is your cell disruption, isolation of DNA, and precipitating the DNA. So in the cell disruption, the cells are grinded or sonicated to remove their membrane lipids, and they are done with the help of detergents. Then we isolate the DNA by removing the proteins with the help of protease enzyme. Then we precipitate the DNA using ice cold ethanol. Then this alcohol is removed afterwards from the palate, and then we have pure DNA in our thing. So here we can see we have a sample. We do the cell lysis, protein degradation, then binding is done, centrifugation, then again elution, then centrifugation. Then you have a pure genomic DNA at the end of your all these steps. So what are the basic rules that you have to maintain while doing so? So if you're taking blood, so first uh, add Triton X100 into the sample. This is a detergent which will helps to lyse your cells. Then wash your cells with specific hemoglobin inhibits reduction enzymes and Taq polymerases. And you should always work on the ice in order to slow down the enzymatic process. Always wear gloves, always use autoclave solutions and store them in the fridge also, and keep all pellets and supernatant until you have DNA that you want. So to get the DNA, cells or lies all in the presence of, so you remove the cell membrane and cell wall with there's no cell wall, only cell membrane. uh, then NaCl is added in order to stabilize the cell, the double helix structure of helix of DNA. Then EDTA is added to chelates magnesium ions and is a cofactor of DNAs which chew up the DNA rapidly. Then we add anionic detergent SDS, which disrupts the lipid layers, helps to dissolve membranes and binds to the positive charge of chromosomal proteins. Then we include a protease ions so that we can digest the proteins that are incubated at the elevated temperature for 4 to 24 hours. And then to remove the protein, we use organic solvent extractions, that is phenol to chloroform. Uh, Phenol is to chloroform 24:1 ratio, and protein are found out at the interface after the centrifugation of 10,000 rpm for 10 minutes for 10 degree Celsius for 10 minutes. Then precipitate the DNA, add 2.5 to 3 volume ice cold 95% ethanol to DNA and leave at minus 20 degrees Celsius overnight. Then centrifuge sample at 10,000 rpm 10 minutes 4 degrees Celsius and wash your DNA pellet to remove excess salts in 70% ethanol and air dry. Then resuspend in sterile distilled water, that is pH 7.4, and store at 4 degrees Celsius or frozen at minus 20 degrees Celsius long term. So to quantify the DNA, to identify how much DNA that you have got, what you have to do is get the amount of DNA that is quantified using a formula. DNA concentration, that is microgram per ml, is equal to optical density that you have found out at 260 nanometer spectrometer, multiply by 100, that is a dilution factor, and multiply by 50 microgram per ml divided by 1000. So double stranded DNA at 260, if you OD, if it is mostly it's 50 microgram per ml, and for the single stranded DNA it's 33, and for the RNA it's 40 microgram per ml. So each unit will have different values. In order to check your DNA purity, it is reflected in the ratio of your OD of 260 to 280, and it should be between 1.6 to 2.0. If it is less than 1.6, then your protein is contaminated. It's been contaminated with protein. If it is more than 2, then the chloroform or phenol contamination is there. Then you have to repurify your sample again. So what we have done so far, we have extracted the DNA from the sample, lyse the cells with that elevated temperature, detergents and enzyme in the salt buffer. Then we remove the cellular proteins and the precipitations of nucleic acids with ethanol, and quantitations and purity measurements of the DNA at the end. So some future aspects. So that's it from this slide. I hope I have shared the slides with you also. Students, give your honest comment on our workshop. How do you like? Would you like to join it for further? Give your comments. We will be happy to know your comments on the. Would you like to join further any workshop? It will be very great to know about your views on that. So, yeah. So let's start with another part. Let's discuss a little bit about PCR also. That is quite important too. Or I can show you something with the isolation of DNA from mouse cells. If there's any virtual workshop present on internet, a good one would be better. No, not this one. This is a sample clip, so we can't see it. This would be nice.
Before beginning any protocol, you want to make sure that your workspace has a proper flow to avoid contamination of samples or reagents. This example is set up for a right-handed student. Tips and pipetters are on the left, racks and samples are in the middle, and the discard bucket is on the right. As your hand moves through the flow, it never unnecessarily crosses over the sample again. This video begins after students have spit into the DNA Genotek collection kits, capped the tubes to release the buffer, and mixed the solution by inversion. At this point, sample tubes should not be labeled to preserve student anonymity during the genotyping process. Using the P1000 micropipetter, 500 microliters is transferred from each spit collection tube into a new microcentrifuge tube. When pipetting large volumes, draw up the solution slowly to avoid bubbles or contaminating the micropipetter. Once the solution is transferred, close the spit tube lid and cap the sample. Repeat this process for all remaining samples. Prior to beginning this protocol, the heat block should be dialed to 50 degrees and turned on by flipping the switch. If it's heating, the light will glow red. Place samples in the heat block. This incubation must proceed for at least 90 minutes, but can also be left overnight. tubes containing 20 microliters of DNA Genotek's proprietary purifying solution, PREP-IT, will be provided. Once the samples have been incubated, you will transfer the entire volume to a tube containing the purifying solution. You may notice that the solution in the tube becomes cloudy. This is normal. Mix the solution with a vortex. To turn on the vortex, press the switch down to touch and turn the knob clockwise to ensure maximum speed. Hold one tube at a time and press the top of the black pad to initiate vibration. Repeat this process for all remaining samples. Embed all samples in ice for 10 minutes. Impurities in the solution will be removed by centrifugation. After flipping the switch in the back of the microcentrifuge, press the open button. Remove the metal lid inside by pulling on the knob. Make sure that the tubes are labeled either with letters or numbers before loading them into the centrifuge. Load samples with hinges facing outward. And balance the tubes across the rotor by ensuring that all tubes have equal volumes. Replace the metal lid and press it until it clicks securely onto the rotor. Close the lid to the microcentrifuge gently. Change the centrifugation time by pressing the arrows to the desired time, in this case 5 minutes. Press the arrows by the speed to navigate to the proper RPM. In this case 14.5 thousand RPM is the max this centrifuge can perform and is close enough to our desired speed. Press the start button. The centrifuge will begin to spin. It is suggested that you watch the centrifuge until it reaches maximum speed to ensure that there are no violent vibrations, which may mean that the tubes are unbalanced or the internal lid has come loose. If this occurs, press the stop button immediately. As the spin is going, obtain 4 new tubes and label them. When the spin is complete, the lid to the centrifuge will pop open. Remove the internal lid and remove the samples gently to avoid disturbing the pellet. Place tubes of the same sample in line with each other to ease the transfer of the solution. After centrifugation, you should be able to see a white pellet that has collected on the bottom part of the tube on the side of the hinge. This pellet contains the impurities in the solution. The DNA is in the supernatant. Using the P1000, transfer 400 microliters of supernatant, which contains the DNA, to the new tube. Place the tip on the opposite side of the tube from the pellet and draw up slowly to avoid disturbing it. Repeat this process for the remaining samples.
Continue the next part.
Precipitate the DNA from solution by adding an equal volume of 100% ethanol to each sample. And mix by inversion 10 times. The solution may become cloudy. This is normal. After incubating at room temperature for 5 minutes, place the samples in the microcentrifuge with the hinges facing outward. Remember to balance your tubes. Spin the samples for 2 minutes. Remove the tubes from the centrifuge gently, being mindful not to disturb the pellet which now contains your DNA. Visually inspect for a pellet at the bottom of the tube on the side of the hinge. A pellet is not always visible, so you can choose to centrifuge again or proceed as normal. In this example, a pellet is only seen in samples C and D, but all samples are carried through the process. Using a P1000, slowly remove and discard approximately 800 microliters of supernatant. Place the tip at the bottom of the tube on the opposite side of the pellet to avoid disturbing it. It is not important at this point that all solution is removed. Just get as much as possible. Expel the tip with the solution inside into the discard bucket. Repeat this process for all remaining samples. Visually inspect the tube to see if this pellet is still present. If it has gone missing, make a note of that. Clean the DNA pellet by adding 250 microliters of 70% ethanol to each sample. Do not mix this solution, as it is imperative that the pellet is not disturbed. Repeat this process for all remaining samples. Place the samples in the centrifuge, being mindful of balance, and spin for 2 minutes. Remove the tubes from the centrifuge carefully. In this step, the pellet still contains the DNA, and it is important to remove as much supernatant as possible without disturbing the pellet. So remove and discard the supernatant using the P200 for precision. This will take 2 or more draws. On the first draw, you can expel the solution into the discard bucket and use the same tip to draw up again, as long as the tip has not touched other surfaces. Repeat this process for the remaining samples. Lay out a few layers of Kim wipes. Open the tube and knock it onto the Kim wipe to remove any large volumes of solution still remaining. Leave the tube open and lay it on its side to evaporate any of the remaining solution from the pellet. Repeat this process for all remaining samples and allow them to air dry for 20 to 30 minutes. If the tube still has spots of solution or if the pellet does not look dry, allow for evaporation to continue. If the pellet looks dry, then resuspend the DNA using water. Add 100 microliters of molecular biology grade water directly onto the pellet side. Wrap the tube and mix by vortexing for a few minutes. Visually inspect the tube to ensure that the pellet has disappeared or has gone back into solution. If it has not, then continue to vortex or allow the tube to sit at room temperature before proceeding to subsequent steps. Repeat this for all remaining samples. Before beginning this protocol, students must fill out the worksheet provided on the last page of this document. This will allow students to calculate the appropriate volume of reagents to mix in the creation of a master cocktail. The purpose of a cocktail is to mix all the common reagents in a multi-sample procedure to avoid excess pipetting and potential errors. In this example of Protocol 2A, 4 students are amplifying the actinin 3 gene from all 4 of their DNA samples. This information is filled out on the left-hand side of the worksheet. On the right-hand side, students use the equations to determine the number of tubes needed and the multiplier for reagent volumes. In addition to the 4 DNA samples, a negative control sample is always included. Therefore, 5 tubes are needed for the PCR reaction. In the creation of the cocktail, pipetting error is accounted for by adding another sample. Therefore, the multiplier for this group is 6. Each row of Table 1 represents a common reagent used in all the samples undergoing a PCR reaction. The multiplier is filled out in all rows of the multiplier column of Table 1. Moving across each row, the volume indicated per reaction is multiplied by the multiplier. "12.5 times 6 equals 75 microliters of red tack added to the cocktail. 11.5 times 6 equals 69 microliters of water added to the cocktail. 0.5 times 6 equals 3 microliters of each of the forward and reverse primer added to the cocktail. Finally, the sum of these volumes should be equal to the product of the final row. 75 plus 69 plus 3 plus 3 equals 150 microliters of total volume in the cocktail. This amount matches 25 times 6. Table 2 is used as a guide for labeling of the PCR samples. As indicated by the equations, this group will be using 5 tubes. The class must collectively number the PCR samples to not confuse samples amongst the groups. In this example, these students have been assigned samples numbered 5 through 9. The sample names are filled out in the second column, and the primers used for each sample are filled out in the last 2 columns. In this example, the forward primer is actinin 3F, and the reverse primer is actinin 3R. The same primers are used for all samples. The common reagents provided are molecular biology water, a forward primer, which you should be sure to check for the marking of PCR to ensure that you are using the appropriate concentration, a reverse primer, and red tack ready mix. The red tack will need to be thawed prior to use, but once thawed, keep the solution on ice to prevent enzyme degradation. Next, prepare the cocktail by using the volumes and reagents indicated on the worksheet. Obtain a new tube and label it as the cocktail. When creating a cocktail, it is best to start with the largest volumes. Though red tack is the largest volume, the solution should be kept on ice as long as possible, so instead the water is added first. When adding each of the reagents, be sure to use the appropriate micropipetter for the volume being added. Next, the primers are added. It is best to pipette small volumes directly into larger volumes to avoid loss of the reagent. Finally, add the red tack and immediately return the stock to ice. Mix the solution and centrifuge all cocktail tubes created by all groups in the class together. Press and hold the short button to do a brief spin.
hope.
The collected cocktail solution will be split amongst the 5 tubes in this example. PCR tubes are provided in strips of 4, so if you need 5 tubes, use 2 strips of tubes. In this case, to amplify actinin 3, 24 microliters of cocktail solution is added to each of the 5 tubes. Before adding DNA to the PCR reactions, make sure that the tubes are labeled. Make sure all DNA samples are mixed before transferring the solution. One microliter is a very small volume, but you should still be able to see a drop in the tip. Put the tip all the way into the bottom of the tube and pump up and down on the plunger before expelling to the second stop. This helps to ensure that the 1 microliter of DNA has been expelled into the reaction. Cap the tubes tightly and flick to mix the samples. Use the mini microcentrifuge with the 8-tube strip head to collect the solution at the bottom of the tube. It is good practice to always mix and collect solutions before every step, especially if the solutions have been sitting for a while. Turn the thermocycler on with the switch in the back. Place the tubes in the thermocycler by pressing the metal latch and lifting up on the lid. Tubes can be placed in any spot or orientation. Close the lid and make sure the metal latch has clicked into place. Heat cycling programs have already been programmed into the thermocycler, so all you need to do is run them by pressing the Proceed button. Use the horizontal arrows to navigate to the program of interest, in this case ACTN, to amplify the actinin 3 locus, and hit Proceed. The heated lid should be enabled by hitting Proceed to prevent extreme condensation at the top of the tube. The thermocycler will ramp to the initial temperature and begin timing the incubation. It is good practice to make sure the machine reaches this step with no errors. After it has initiated properly, the machine can be left while the reaction proceeds for several hours. Once the run is complete, the cycler can be turned off by simply flipping the switch in the back of the machine.
So we are done with the DNA isolation. We're done with the PCR, and so we have understood the basics idea behind our molecular biology, um, basic tools behind them. So I hope this PCR, what we have done, I will just skip this part, not speak much. So as we have discussed, to have further thing, some history is there, purpose we know. So for the denaturation is done as 90 to 94 degrees Celsius, as we've seen in the video, and the double-stranded DNA melts away in them and giving rise to, at 92 degrees Celsius they got separated, single-stranded, and then annealing is done from 50 to 70 degrees Celsius. And 72 degrees Celsius, with the help of Taq polymerase, DNA polymerase, you anneal these primers and extend the DNA at 3' end of the chain. And products of extractions are available, and we are good to go for the reaction. So these are the chemical components as we have discussed required. And then yeah, some application, these instrumentations we have discussed. That's it. So PCR is almost being self-explanatory, explained in the video, so I didn't go in details about that. So this is PCR. Now next is this, I have shared you already, right? The introduction. Yes, this has been shared. Now I want to take you to the next part, that is types of DNA. So as we all know that there are mainly of 4 types of DNA that are present. That is According to Watson-Crick model, so they said mainly bDNA or B form, but in different conditions, they could be aDNA, zDNA, cDNA, ddNA, and eDNA. And this division is formed based on the structural diversity. So this is the according to the X-ray analysis of DNA crystals at atomic level, we have revealed that DNA exhibits various varieties. So bDNA, which is mostly common, around 92%, whereas the aDNA was found with a relative humidity of 75%, and the zDNA is a left-handed. That's exceptional DNA. Most of the DNAs are the right-handed ones, coiled around your axis, but the zDNA is left-handed, which winds to the left in zigzag pattern, with the cDNA formed at 66% relative humidity and in presence of lithium and magnesium ions. So ddNA rare variants with 8 base pairs per helical turn form in structures devoid of guanine, whereas the eDNA extended or eccentric DNA. So basically it's 6 types of DNA so far. That's the basic structure of a DNA. We can see with the minor groove and major groove. The bDNA was first described by Watson and Crick, commonly found in DNA, and they consist of 2 helical polynucleotide chains coiled around common axis, and 2 helicals are wound in such a way to produce 2 interchain spacing or groove. Further, they have this major and wide groove, minor and narrow groove, and these grooves prove surface with which proteins chemical drugs can interact. So this is the diameter of DNA, 20 angstrom or 2 nanometer, and one complete turn is around 34 angstrom. A major groove and the minor groove are there with a sugar phosphate backbone and the nitrogenous base pairs, and the distance between your each nitrogenous base pair is 3.4 angstrom. So that's the B form, that the normal DNA that we see in our body also, 2 helical wind along the molecules, 2 chains going anti-parallel, right hand twisting, uniform diameter of 2 nanometer, complementary base pairing, around 10.4 base per turn, and rise per base pair is 3.4 angstrom. This is aDNA. The aDNA is the one with a possible double helical structure, which can adopt along with the other biological active helix structures. And the right-handed double helix structure, short and fat compared to bDNA, occur only in dehydrated sample of DNA, such those used in crystallographic experiments. So aDNA was originally identified by X-ray diffraction analysis of DNA fibers at 75% relative humidity. The grooves are not as deep in bDNA. The bases are more tilted, and base pairs per turn is 11, and the rise per base pair is 2.3 angstrom. zDNA, this is one of the main possible DNA double helix structures. So this is the one with the exceptional left-handed double helix structures, wings to the left in zigzag manner. So they termed as a zDNA. They were discovered by Rich, Nordheim, and Wong in 1984. It has an anti-parallel stance as a bDNA, and it is a long thin as compared to the bDNA. So this is the main difference between A, B, and Z. Helix is right, A and B, Z is left. Width is the widest in the A, this is the intermediate, Z is the narrowest. The plane of base is perpendicular to the helix axis in A, perpendicular to the helix axis, both, it's the same in all three. The central axis is 6 angstrom holes along the helix axis. This is tiny central, no internal space, and the major axes are narrow and deep, wide, deep, no major groove here. So that was a little bit about types of DNA. Mhm. Now we have to study about DNA replication. So I didn't share with you these beautiful PCR techniques.
Before beginning any protocol, you want to make sure that your workspace has a proper flow to avoid contamination of samples or reagents. This example is set up for a right-handed student. Tips: precipitate the DNA from solution by adding an equal volume of 100% ethanol to each sample. The collected cocktail solution will be split amongst the 5 tubes in this example. PCR tubes are provided in strips of 4, so if you need 5 tubes, use 2 strips of. Before beginning this protocol, students must fill out the worksheet provided on the last page of this document. This will allow students to calculate the appropriate volume of reagents to mix in the creation of a master cocktail. The purpose of a cocktail is to mix all the common reagents in a
So you can
multi-sample
watch these
procedure
videos
to avoid
anytime
excess pipetting
from
and
this
potential errors.
batch here.
In this
This
example
is
of
part
Protocol
1, part
2A,
2.
4 students are amplifying the actinin 3 gene from all 4 of their DNA of tubes. In this case, to amplify actinin 3, 24 microliters of cocktail solution is added to
Okay,
each of the
all
5
good.
tubes.
So now let's go to the next. Yeah, DNA replication. Before that, I just want to share something about central dogma. Some quick slides with that. That central dogma, as we have discussed, mostly we say that DNA makes RNA, RNA makes protein, but this is not the original meaning. So the first stated statement was given by Francis and Crick in 1957, which were published in 1958 about the central dogma, which states that once information has passed into the protein, it cannot get out again. So in more details, the transfer of information from nucleic acid to nucleic acid or from nucleic acid to protein may be possible, but transfer from protein to protein or from protein to nucleic acid is not possible, is impossible. So in general, the blue articles, as we have discussed, DNA could self-replicate, produce RNA, then protein. RNA is special case, which leads to go back and produce DNA. So what the Francis and Crick Maybe this was the major part that they have discovered. The three major classes they had divided, that is the general transfer, special transfer, and unknown transfer. So they divided this your what you say the central dogma into three different categories. The first category is the general transfer, in which your DNA is copied to DNA, that is DNA replication. DNA is went into the mRNA, that is transcription, and then further into the mRNA is transferred to the protein, that is a translation. And in the special transfer, mainly RNA could be copied from RNA, RNA replication could happen, and then with the help of reverse transcriptase enzyme, reverse transcription could happen in order to produce complementary DNA from RNA. The unknown transfer are also there, which shows that protein could be copied from protein, synthesis of RNA using the primary structure of protein as a template, or DNA could be synthesized using the primary structure of protein, and these are not naturally are occurring. So some exceptionals are there, unknown transfer. Just want to discuss about this little bit about central dogma. Mhm. Okay. Um, So this is also very important part, that DNA act as your genetic material. How DNA could be actually your was told that it's a genetic material. So I will take you to the 12th class history classes, which we might have forget, with Hershey and Chase for experiments, Avrold and TKC experiments, and Melson experiments, in order to find out what is the reason behind our genetic information. So before we discuss the evidence that from experiments to prove that genetic material of most living organisms or viruses is double-stranded DNA, let's first review the James Watson and Crick, what they have discovered about the structure of DNA in 1953. So earlier, the Mendel was the first one who set these genes as effectors, which was not discoverable before. So one gene, one enzyme theory was postulated, which control the structure of proteins, and genes are known to be carried on chromosomes. And the chromosomes are found to consist of DNA also. And the discovery of DNA as a genetic material. So one of the first study that ultimately led to the identifications of DNA as genetic material was done by Frederick Griffith, involving the bacterium Streptococcus pneumoniae in 1928. And this bacterium, which causes pneumonia in humans, is quite lethal in mice also. So Griffith experiments came into play role in order to find out the smooth appearance of colonies, which he labeled them as S, and the rough colonies as R. So we will discuss about them in details soon. Um, so yeah, the Griffith experiment, that is your transforming, it's called also as transforming principle. So he conducted experiments to show the transforming principle in bacteria. So basically what he conducted, that experiments on mice and pneumonia, and bacteria Streptococcus pneumonia, and he found two strains, that is virulent and non-virulent. So the virulent were the smooth strains and the non-virulent were the rough strains. So S strain bacteria produce capsule and is pathogenic, and the R strains lacks capsule and is non-pathogenic. So we have rough strains. These are on the left hand side, most left hand side, are non-virulent, which doesn't have any capsule around, and when they go into the mouse, they are not bad to it, and mouse lives. And there is a smooth strain which are virulent, which having a capsid around it, so they kill the immune system and mouse dies. But when we add heat killed smooth strains, so this is the heat killed smooth strains, when we add into the mouse, then it starts to live. So the function of the deleterious function of the virulent has been killed because of this. But when we add heat killed smooth strain and rough strain, what we see, the mouse dies again. and that was quite strange. The reason he told that the rough strain got transformed into the virulent form. The DNA of your rough strain got changed and leads to the death of the mouse. But it was not told that what was the reason behind it, what was the reason that mouse was dying. So it was not so convincing experiments. So I will repeat all these things. So when the R strains are injected into the mouse, it is non-pathogenic and does not cause pneumonia, and the mouse continue to live in that. But when the S strains are injected into the mouse, that causes pneumonia and mouse dies. But when we add this heat kill strains and we inject it into the mouse, that does not cause pneumonia and the mouse continue to live afterwards. And when the heat kill S strains and R strains are mixed, injected into the mouse, so that cause pneumonia and mouse dies. So in conclusion of these experiments, R strain bacteria had been transformed by the heat kill S strain bacteria, and the transformation of R strain to S strain is due to the transfer of genetic material. And the biochemical nature of genetic material was not defined from his experiments. So then, to understand the biochemical nature, Oswald Avery, Colin McLeod and McCarthy, they three together did experiment from 1933 to 44 to identify the transforming principle of Griffiths to find out the biochemical nature. So what they did, they purified these molecules, that is your protein, DNA and RNA, from your heat killed S strains, which are non-virulent, and they added digestive enzymes of each into them to see which will transform to the R cells to the S cells. So these are the, yeah, so heat killed S strains, they added protease plus live R strain, so they transform the R cells to S strains in this case, and they added S strain with RNAs with live R strain, both together they also got transformed. But when they added DNAs, they were not able to transform. So main reason is behind that the DNAs will kill all the DNA that is being given to the live R strain and transforming it, but RNA and protein were not playing role in that. So that's the reason DNA was a biochemical thing that were transforming, making your non-virulent R strain to virulent R strain from your heat-killed R strain. So that was the basic concept behind it. So in conclusion of the experiments, the proteins of the heat-killed R strain is not the genetic material. So RNA of heat kill S strain is not the genetic material, and DNA of heat kill S strain is the genetic material. So because DNA digested with the DNAs mixed with the R strain, which were unable to transform R strain to S strains, but all biologists are not convinced because of this. Then the genetic material is DNA. This DNA is a genetic material which is proved by Alfred Hershey and Martha Chase in 1952, and they worked on the virus that infects bacteria called bacteriophage. So what they did, see this bacteriophage, they go into the virus, they transform it, and they come out after the infection, infecting it. So they want to check whether the DNA is a genetic material. In order to do so, the viral genetic material become part of the bacterial genome, and it manufactures most virus particles using host content. So Hershey and Chase worked to discover whether it was a protein or DNA from the virus that entered the bacteria. So they did this blender experiment. So they grew some viruses on media having the radioactive phosphorus and some other on the radioactive sulfur. So one which were grown on the phosphorus have radioactive DNA but not radioactive protein, because phosphorus present is in DNA, not in proteins, whereas the sulfur is present in protein, radioactive protein, but not on the radioactive DNA. So based on that, they did this experiment. So in the first case, we can see protein codes of the phages are radioactively labeled, and then phages infect bacteria with genetic material, and the no radioactivity enter to the cell. And in the second one, DNA of phages is radioactively labeled, and phages infects bacteria with genetic material, and radioactivity enters into the cell. So that means once your phages, the protein codes are being, in this one the sulfur, so mainly they didn't enter because it was a protein, but once the DNA was infected with phosphorus, they got enter into the body of bacteria or plasmid phages and leads to the infection. So radioactive phages were allowed to infect E. coli bacteria, and the phages transfer the genetic material to bacteria also, and the viral coats were separated from bacteria surface by blenders, and the virus particles were separated from the bacteria by centrifuge machine. The things that we observed were where the viruses had active radioactive DNA were radioactive, no radioactivity in the supernatant, but bacteria infected with the viruses had radioactive proteins were not radioactive. So in conclusion, the experiment was that infective agents that made the bacteria radioactive, hence DNA is the genetic material, not the protein. Then comes replication of DNA, which is a process your DNA produces exact copies of original DNA. So in eukaryotes, DNA is a double-stranded and two strands are complementary to each other. So this DNA replication is actually a semi-conservative method. So it is the most common method of DNA replication. So it takes place in the nucleus where the DNA is present, and the replication takes place in the S phase of the S cell cycle. And deoxyribose nucleotides needed for the formation of new DNA strands and are present in the nucleoplasm. At the times of replication, so there are two strands of DNA which are separating. There's a new, this is a new strand, and these are the and then these are the old strands are going on, and the new strands are correspondingly being produced here. So each strand acts as a template for formation of new strand. A new strand is constructed on each old strand by complementary base pairing. Hence, two exactly identical double-stranded DNA molecules are formed. So in each new DNA molecule, one strand is old while the other is newly formed. Hence, Watson and Crick describe this method as a semi-conservative replication. So further, this Mansell and Franklin, they did this to understand this semi-conservative nature of our DNA. So Matthew Massillin and Franklin start the experiments. So what they did is they got the E. coli, yeah, and the DNA becomes uniformly labeled with the 15N nitrogen base pair. So E. coli grown in the 15N labeled medium like this, nitrogenous, and they grow it into the culture with a zero generation, and then they have added this 15N labeled E. coli into the 14N medium. I think this is not better, this is better experiment. 15N containing medium, nitrogenous in E. coli culture, and the CS cesium chloride gradient. So DNA composition is heavy, 15N 15N DNA. When we see in the DNA bands, this is the one band, and in the densitometric scans we see this 15N 15N DNA here. But once we replicate them in the N14 media, 14N media, so what we see is that it's a mixture of both. Yeah, it's intermediate density with both 15 and 14N, and we see between bands between in between, and also we saw the densitometry scans in between 15 and 14N DNA intermediate. Then we keep growing it for second replication. So what we see is that the one cycle is with 15 and 14N, and other is your 14 and 14N. So that means it's a semi-conservative, and we see these two peaks and once they grow also, so this one will give mainly 14 and 49, and this one will give again two different 14 and 59. So this proves that your DNA replication is a semi-conservative in nature, and the results that you discovered because of the DNA replication. Same things have been discovered. So this is the explanation of the same experiments, that they grew E. coli on 15 as ammonium chloride culture medium, where 59 is a heavy isotope of the nitrogen, and the both strands of DNA have 15N, and these bacteria are shifted to 14 NH4 solution culture media, and they're subjected with the cesium chloride density gradient. And the hybrid intermediates were found out, and the next generations equal the amount of the DNA, and hybrid DNAs are formed. So that's it. I think we have covered lots of topic today. We have done some practical part also. We have done some theory part also. So all in all, we are going good, good on time. So the last 10 minutes are for the question and a Q&A. If you have any questions, you are feel free to ask me. I will happy to answer your questions. So from your this perspective, haven't done DNA replication yet, but I have taught you some extra material that about the nucleic acid. Nucleic acid is a genetic material which is a broad subject itself. We have discussed types of DNA, so we still have to discuss about DNA repair system in prokaryotes and eukaryotes, and DNA replication system. But beyond that, today we have discussed about PCR. How does we do that in virtual lab? We have done the DNA isolation. So if you want, we can also go for more molecular biology techniques, more focus with the chromatography. So tomorrow maybe we can focus on that, until and unless we are done with the subjective part also. So we can do some mixture of things. So no worries. So I'm open to your questions. If you have any questions, feel free to ask me. I will be happy to answer your questions, queries. This file is not going. I don't know why. So we should remove this file. I delete this. Oh. Okay, so that was the basics for today. And students, I have shared you one, give your honest comments, send us the screenshots. I've sent, yeah, this EduFabrica workshop's helpful. And we're also having quite the same workshop that we're having the mini ones. There is a proper training where we have of all these workshops. We have a lot of workshops actually going on in the biological series. We will teach you the, what do you say, review writing articles, how to write review articles. So this internship letter you will get from us. So just think about it. Let us know how you want. Because if I start any topic now, so I cannot cover. So you might be having another classes also in next 10 minutes. So I cannot make it happen. Okay, if not, we can end up our session here maybe, and then we see us tomorrow, same time, 5 p.m. and we continue with the further topics. Maybe I try to bring more virtual workshops there for you. Okay, thank you. Take care. Stay safe.